Thank you, Alice, very much for the introduction and also for the invitation for this webinar. And I would like to use this opportunity to present one of our recent studies that focuses on amino acid auxotrophies in the human gut microbiome. But before I start with the topic itself, I want to say that this is a study that was mainly performed by two group members from my group here in Kiel. This is Svenja Starke and Daniel Harris. They have contributed most of the work to that study. So first of all, maybe this term auxotrophy might not be familiar to everyone, so I will give a brief definition here. An auxotrophy means that is the inability of an organism to grow without this vital compound in its environment. So these compounds could, for example, be amino acids or vitamins, and these auxotrophic organisms, basically they lack the biosynthetic pathway in order to produce these metabolites by themselves, so they rely on the external supply of these compounds. So the opposite term for auxotrophy is prototrophy, which means that the organism can produce that nutrient that is required for growth. You can kind of picture microbial auxotrophies very similar to the essential amino acids to the human organism. So we know that some amino acids cannot be produced by humans, so we need them to obtain them from the diet. And now the question that we were asking here is, how does it reflect it when we look at the organisms that live in our gastrointestinal tract, which are the bacteria and archaea mainly that we have analyzed in the study? What are essential amino acids for these organisms that are so closely associated to us? So we addressed four main questions in that study. So the first one is, how common are amino acid auxotrophies in the human gut microbiome, and what is the variation between individuals? This sounds very basic, but when you think about auxotrophies and look into the literature, there's a big knowledge base on auxotrophies in other environments, for example, in freshwater environments or in soil communities. And there we know that auxotrophies have a huge impact on the dynamics of the microbial community and the metabolic contribution to that environment. But for the human gut microbiome, this was not known before, or how these auxotrophies are even represented in the human gut microbiome. So the second question, if there are certain associations between the gut microbial auxotrophies and host metabolism, then my students also ask the question, what are potential sources of these amino acids that these auxotrophic microbes require from their environment? And a very general question that we discussed in this paper is, how do auxotrophies affect gut ecology in the human gut microbiome? So here we'll briefly present the study design or the workflow that we applied in the study. First, we started by analyzing genomes from the human gut microbiome. So there are plenty of projects which try to summarize or collect all microbial genomes that are out there that have been sequenced already for the human gut microbiome. And for this study, we used the so-called HRGM collection, which means the Human Reference Gut Microbiome. That was at that time the largest catalog of gut microbial genomes that we could use in that study. Next part is a very computational part of our work. And here we used a software called GapSeq in order to reconstruct the metabolism or the metabolic network of these gut organisms, these microorganisms. And here the idea is to have in silico representations of these microorganisms in order to make predictions about their auxotrophies and prototrophies. I want to give you a brief introduction as a nutshell here to show you how that works, this prediction. So we start with the annotated genome that we got from the genome catalog. From the annotation of the genome, we know which enzymes are encoded in the genome. And by looking into databases, which reactions can be catalyzed by those enzymes, we can collect all the reactions that the organism is capable of catalyzing in theory, at least. And when we put together all these reactions in one network, we come up with the so-called metabolic network or genome scale metabolic network, which is a representation of all the catalytic capabilities of that organism so far as we can know this. And this metabolic network is then represented as a so-called stoichiometric matrix, which is just like a computer way of saving the topology of that metabolic network, and which we then can use in order to make predictions about the microorganisms' phenotypes. For example, the growth rates or growth fields of these microorganisms in certain environments. We can predict metabolic byproducts and cell maintenance mechanisms. and we can also predict the impact of specific nutrients on the organism's metabolism. For example, we can predict which metabolites or nutrients are essential for that organism in order to survive or to reproduce in an environment of the human gut. We use these metabolic models to predict these auxotrophies, and the way of doing this is actually quite simple. So we took the in silico microorganisms that we have now as a representations of their metabolism, and we asked, for example, can this organism grow with tryptophan, and can it grow without tryptophan? And usually with the amino acids, of course, they can grow, but if we take it away, some of these organisms might show no growth in the computer predictions, whereas others can grow. And the ones that can grow without tryptophan, they are then coined prototrophic organisms for tryptophan, and the ones that cannot grow without tryptophan are termed auxotrophic. So now you might say, okay, these are all computer predictions, but how can we be sure that these predictions are reliable? So we try to find as much experimental data on auxotrophies in human gut microorganisms as a validation approach, and here we could collect the information about auxotrophies that have been experimentally verified for 36 gut isolates of gut microorganisms, mainly bacteria. And we were happy with the results because we got an overall accuracy of 93%, a specificity of over 95%, and the sensitivity, so the true positive auxotrophy predictions of more than 75%. This gave us some confidence that we can rely on our predictions in order to estimate also the distribution of auxotrophies in the whole human gut microbiome. In this validation approach, we have also included 127 genomes from known prototropes. So these are microorganisms which have been cultivated in minimal media in the laboratory, and these media did not contain any amino acids. So this is a confirmation that these are prototrophic for all amino acids. And when we predicted these auxotrophies for those organisms where we hope to not see any of the auxotrophies predicted, we did not predict too many auxotrophies here. So it's a false prediction. We made less than 1% in this data set. So then we wanted to see how is the distribution in the human gut microbiome. And as you know, the composition of the human gut microbiome can be very person-specific, or is always very person-specific. And in order to assess this variability of auxotrophies and their distribution, we analyzed 3 different cohorts. One cohort was used from directly in this study that I present here. And we have also added 2 additional cohorts in order to see if their predictions in one cohort are also seen in the other cohorts. This cohort from this study is from Kiel and Kiel area, so northern Germany. The same is true from this Troji et al. Study, but a bit earlier. And this larger study here from Shen et al., this is from the northern Netherlands, and this is data we got provided by Lifelines in the Netherlands. So we use these data from the metagenomes to quantify the abundance, the relative abundance of these microorganisms that are in the genome collection. And by combining this with the information about the auxotrophies that we have predicted, we were able to estimate the auxotrophy frequencies in all of these 3 cohorts. And this is shown here on the x-axis, the amino acids, on the y-axis, the relative abundance of auxotrophies in this one cohort of Kiel from Kiel. And as you can see, the amino acids that are also essential to the human host are also very frequently essential to gut microbes in the microbiomes of individual persons. So, for example, tryptophan, that was more than 50% of the microbiome was needed tryptophan in order to grow. But also other amino acids that are essential to the human host were quite frequent in the auxotrophies in the human gut microbiome. Whereas the non-essential ones, amino acids, they are less frequent as auxotropes in these environments. One thing we wanted to analyze here is the impact of auxotrophies on the ecology of the human gut microbiome. And here we wanted to see, okay, how does the distribution of auxotrophies relate to the overall diversity of the human gut microbiome? And here the diversity is measured as the Shannon index on the x-axis, and on the y-axis we have this measure which we call the abundance weighted average of auxotrophies. And you can picture this as the average numbers of amino acids that the gut bacterium cannot produce. And this is then weighted by the relative abundance of that specific organism in these microbiomes. And as you can see, we observed here quite strong positive correlation, meaning that if we have more auxotrophies in the microbiome, then also the overall diversity of the microbiome seems to be higher. And this was measured across all amino acids, but if we look also into individual amino acids, the same was true for almost all these amino acids that have been tested in the study. Another measure that the Svenja came up in our study here was to ask, okay, how much can we infer from the auxotrophy distribution about cross-feeding that might take place in these microbial communities? And cross-feeding is the process where one organism is producing a certain metabolite that is then consumed by another cell. And of course, auxotrophy in our microbiome, we have always auxotrophs and we have prototrophs. And the idea is that maybe some of these amino acids that are required by the auxotrophs are actually produced by other members of that microbial community. So there's a cross-feeding from prototrophs to the auxotrophs. And in order to quantify this potential of cross-feeding, Svenja came up with this approach to calculate the Hamming distance or the average Hamming distance. And this is by looking at different organisms that coexist in one of these environments, for example, species A and species B. We made the predictions about the auxotrophies, and then we simply calculated the numbers of differences from the prototrophs and auxotrophs predictions. Here in that other example, there's for tryptophan, one is prototroph and one is auxotroph. And this means that we have a Hamming distance here of 1, which means that there's one difference in the amino acid predictions, in the auxotrophy predictions. But for others, it could be more. For example, for this example here, species A is auxotroph for leucine, tryptophan, and alanine, whereas the others is only auxotroph for tryptophan and isoleucine, and there we get a Hamming distance of 3 because we have 3 mismatches of these predictions. So these Hamming distances is a measure of potential cross-feeding between the species that coexist in an environment. And so we wanted to see if that also correlates or kind of associates with the diversity of the microbiomes. Again, on the x-axis here, we have the Shannon index. On the y-axis here, the average Hamming distance, basically the measure of potential cross-feeding. And here, that correlation was even stronger, meaning that with higher diversity, there seems to be a higher metabolic complementarity between the coexisting genotypes in the environments. The next question was if these auxotrophies that we predict also associate with certain host metabolites, because the different auxotrophy profile in the human gut microbiome could indicate different metabolic processes, especially in amino acid metabolism. And we wanted to see if that is correlated with certain metabolites in the host metabolome to see if there are kind of interactions that are depending on the distribution of auxotrophies. And first of all, we here in that metabolome data set that we have generated with the biocrates kits, we did not see, surprisingly, any associations of these auxotrophy frequencies in the gut microbiome and the serum levels of amino acids. There can be many reasons for this, why there's no association, but maybe this is already telling us that amino acids from the host are not very frequently the sources of amino acids for the microorganisms that the auxotrophic microorganisms that reside in the gut. But we saw plenty of different positive and negative associations of metabolites, mainly with metabolites that are already from previous studies known to be of microbial origin. For example, we had this IAA or IPA, which we were also introduced earlier here by others, but also with TMAO and hippuric acid. So this tells us that there seems to be a link of the auxotrophy distribution in the human gut microbiome is a contributor to the human metabolome, especially with compounds that are derived from amino acids, for example, the IAA. So one next question that we wanted to ask, since we had already this very interesting association between the diversity and the distribution of auxotrophies, we wanted to see if also auxotrophies and the frequencies relate to the stability of the microbiomes. And here we could use, we were able to use the two other cohorts from our study. So one from Kiel area and the other study from the Northern Netherlands. And these cohorts are interesting because we had here longitudinal stool metagenomes, one from baseline and 3 years after the first collection for the Kiel cohort and for the Netherlands cohort after 4 years of the initial stool collection. So these two cohorts were quite comparable from their setup, but they are geographically distant, so we could see if the associations with the stability of the microbiome is also reproducible across different locations. And what you can see here on the y-axis is once this abundance weighted average of auxotrophies, so what I've introduced earlier, and the average Hamming distance as a measure of potential cross-feeding in these communities for amino acids. And here on the y-axis are the microbiome stability, which we have calculated as 1 minus this beta diversity measured beneath that. So meaning with a higher value of this microbiome stability, meaning a higher stability. And interestingly, we found a positive correlation here in both cohorts. For example, here in orange, the Kiel cohort, and in purple, the Netherlands cohort. And in both, we see this positive correlation that with higher auxotrophy frequency and higher potential of cross-feeding of amino acids, there seems to be a stronger stability of the microbiome over this very long period of time, which is 3 or 4 years. So cross-feeding, like I said, could be one source of the amino acids for the auxotropes, but a more intuitive source of the amino acids is obviously diet. So in one of these cohorts, we were also able to get dietary information from diet questionnaires. So giving us the estimates of how many grams of amino acids a person is consuming per week. And that we were able to correlate with our predictions of auxotrophies. Here, the hypothesis was that if there's a large intake of a certain amino acid, then we would also see more of the auxotropes and the gut microbiome. But for our surprise, we did not see any statistically significant correlation here, which could also be, of course, due to the lack of power or kind of also the lack of accuracy of these dietary questionnaires. But this data at least did not give us any evidence that diet is a contributor to the distribution of auxotropes in the human gut microbiome. So in summary from that study here, from first questions of how common are auxotrophies in human gut microbiome, we can say that they are very highly prevalent across different cohorts and across different countries. And we also observed that tryptophan has the highest frequency for auxotrophies in the microbiome. And we have also seen associations with the host metabolism, especially with bile acids and indole derivatives or tryptophan derivatives. And from our data that we have, we did not have any evidence that diet is a main contributor to the frequency of auxotrophies, leaving the other factors, cross-feeding, for example, or amino acids that are derived from host resources, that these are more likely the main sources of amino acids for these auxotropes. And our data suggests, especially with the Hamming distance that we have analyzed in that context, is that cross-feeding based on between auxotropes and phototropes could be a contributor to the microbiome diversity and the maintenance of this diversity, which is then reflected in the long-term stability. So with that, I come to an end. I want to, of course, acknowledge my whole group, especially Svenja and Daniel, who have contributed most to this work, but also the clinician scientists involved in the study, which were very open to address also these very ecological questions that we have addressed in the study. And of course, also Sven and Schubert from the ITEM in Hannover, who performed the metabolomics analysis for us. And thank you for your attention.
Thank you very much, Silvio. You made it very clear and understandable, even for people like me who are more on the metabolomic side. So I feel like I learned a lot about your work. Thank you so much.